So in this video, we're going to look at mutations. We covered this in AS, so it's just really a revision looking at the types of mutations and their effects. So for types of mutations that we've come across: substitution, addition, deletion, inversion, and translocation. We're going to look at each of those in turn. First one is substitution, which is when you replace one base with another one. So for example, if I have a DNA base sequence like that, with obviously its pair, like so, its complementary base pairs on the other DNA strand. So there's one DNA strand, there's the other DNA strand with its phosphate sugar backbone. These bases held together by hydrogen bonds, like so. And when the DNA is copied for some reason, there is a change. So we're going to make a change here. We're going to put a T in instead of an A. And the rest carries on as normal. And the other pairing complementary base will also, as a result, change. And so we have a change, a substitution of this base over here. So that's one possibility. Now that could be silent, i.e. it will have no effect on the phenotype, and we'll see why in a minute, or it could have an effect. So let's think, why can the mutation be silent? And that's what we're going to start on. So if we look at silent mutations first, the first reason is the idea that the code is degenerate. So we know that in DNA, the code, the genetic code, is in triplets, like so, each coding for a specific amino acid. Now, we also know that in order to make a triplet, you've got 4 choices of base for the first one, A, C, T, and G, 4 for the second one, 4 for the third one, which gives us 64 possible combinations of bases, but we only need to code for approximately 20 amino acids plus some stops, codons. So therefore, with the code is what we call degenerate. In other words, more than one triplet codes for the same amino acid. So in other words, you might have a mutation, but you're not going to be aware of it because you've got the same amino acid coded for. We all have mutations that we are not even aware of. So that's the first reason. Second reason is that you put a new amino acid in, but it's not involved in the bonding between the R groups in the tertiary structure. So if we remind ourselves of our bonding, here is our polypeptide chain, our primary structure. I'm just going to put that for primary. That folds to form either an alpha helix or beta pleated sheet, called our secondary structure. And that is held together by hydrogen bonds only. And they are nothing to do with the R groups. They're to do with the carboxylic acid and amino groups. And then that one will fold itself up like so, giving what we call our tertiary structure. And that is held by the bonding between the R groups. And this can be either ionic bonding or hydrogen bonding or disulfide bonding. So let's remind ourselves what we mean by R groups. If we think about an amino acid is made up of an amino group, a carboxylic acid group at the other end, and in the middle carbon, you have a hydrogen and then the R group. And the R group is what makes each amino acid unique. So every amino acid has an amino group. Every amino acid has a carboxylic acid group, but different amino acids have different R groups, and that controls which of these 3 bonds they make and the structure, therefore, of that tertiary protein. So if the amino acid is not involved in that R group bonding, then the tertiary structure will stay constant and it will not change. There is, of course, another possibility. It's nothing to do with the actual amino acids at all. In fact, new amino acids are put in, but it's a recessive characteristic. So because we have 2 alleles, one on each chromosome, if one allele has mutated, but the other allele is still normal, then it may have no effect at all. So what if it isn't silent? Well, possibility number one is that you could get a new amino acid to be inserted, obviously. And if that amino acid is involved in the R group bonding, then the tertiary structure will now change. So for example, the active site may no longer be specific and complementary to the substrate, or the binding site on a hormone may no longer be complementary to its receptor, whatever it happens to be. Sometimes a substitution could cause a stop codon, and that is going to shorten the polypeptide. So if normally the polypeptide would have gone on for a long time, then maybe a mutation could introduce a stop codon, getting a very short protein. Just an example here, you don't need to remember it, but sickle cell anemia, which if you don't know, makes the red blood cells sickle-shaped in low oxygen levels, and that gets stuck in the capillaries. Sickle cell anemia is caused by a single base change in the hemoglobin, and that single base change of an A to a T causes valine to replace glutamic acid, and that causes the hemoglobin structure to be incorrect, and that's what causes these sickle shapes to the red blood cells. So addition and deletion are the next ones to think of. So an addition is simply the insertion of a new base. So I have a sequence, CAT, AGC, and I literally insert a new base in. So now I've got CAT, and I've still got the AGC, but in between that, I've now inserted another T, for example. Deletion is the removal of a base. So following the same sequence, CAT, AGC, I now remove the base. So I've got CAT, I lose the next A, and I've got GC afterwards. Now, the problem with this is they're what we call frame shift mutations. So let me explain what I mean by a frame shift mutation. So let's look at the insertion one we had. So we had CAT, AGC. That was coding for one amino acid, that was coding for the next, and then the next lot of 3 will code for the next one, and the next lot of 3 will code for the next one, and so on. If, however, I now put in my extra base, it was here, then I've got the first triplet unaffected. The second triplet is now completely different, and so is every triplet afterwards. And that will be the same for a deletion. If I have a deletion, so I've gone, I had before CAT, AGC. I'm going to put in another few bases just so you can see. 1 triplet, 2 triplets, 3 triplets, and so on. I remove one of them now. So I've got the C, the A, the T, the G, the C. Now everything just shifts up by 1, like so. Keep going. First triplet not affected. Second triplet is now different, as is the third, as is the fourth. Okay, so every single triplet after my mutation is now different, which means I get a whole new polypeptide chain, a whole new amino acid sequence. Okay, so all the triplets are then affected. Whole amino acid sequence is therefore affected. Okay, what is inversion? So inversion is when 2 or more bases are switched order. So for example, CAT, we got a switch of bases there. So these 3 all switch order. Okay, so the T stays in the middle, obviously, but the G, the T, and the A have been inverted. And there you can see very quickly, we've got one amino acid, which is coded for by CAT, another which is coded for by GCA. Again, we've completely changed the 2 triplets. We won't necessarily have an effect on any other triplets, but those 2 triplets are completely different, which means if those 2 amino acids are important in the bonding of the R groups, in the tertiary structure, we will have a new tertiary structure, which in most cases would be a disadvantage. Obviously, natural selection means occasionally These mutations give us a selective advantage, but in most cases, it would be a selective disadvantage, okay? So it's likely to affect at least one triplet and possibly more. Translocation is the last one. Translocation is when a section of bases is moved from one locus or place on the chromosome to another. Now, this could be the same chromosome or another. So you literally, if you imagine you have a chromosome here, you cut off one section, could be in the middle, and you literally move it to another place. So it could be there, or it could be moved to a completely different chromosome. So not chromosome, complete different chromosome number, for example, and put on the end of that, like so. So an example of this is Down syndrome. One form of Down syndrome is obviously trisomy 21, 3 copies of chromosome 21. But in another example, parts of chromosome 21 can be translocated to chromosome 14. And what can happen, therefore, in the if this happens during the production of the sperm or the egg, you could end up in the embryo with 2 chromosome number 21s perfectly normal, 2 chromosome number 14s, but one of the chromosome number 14s also has an extra bit of chromosome 21 stuck on the end, and that can cause Down syndrome due to having 3 of certain genes. The severity of the Down syndrome is not as bad as having trisomy 21, and these children born with Down syndrome like this will also be fertile, so could pass on the Down syndrome to their children. So the translocation can have a really dramatic effect. It will have a dramatic effect if a particular gene is interrupted. So if you cut within a gene, so for example, if I had, imagine a chromosome and I have a gene here, and for some reason in my translocation, I cut sort of the middle of the gene out and put it somewhere else, then obviously that's going to have a real problem. Or if it happens in meiosis and the zygote ends up with 3 copies of the genes, which is the example we've just started looking at. So this is the summary of all of the types of mutations that you come across and whether they are silent or not and the possible effects they can have.